and some of you may know me as Han Hongxue because I forgot to change my name to the school. So today I'm going to present a paper about stimulated gamma delta T cells increasing the in vivo efficacy of trastuzumab in HER2 positive breast cancers. So I will give background information first, and step by step I will introduce.
You should slow down. You have over 1 hour.
That's way too much. Okay. So step by step I will introduce the motivation behind this experiment and this research paper and the reason the experiment is being conducted. So I will start with the introduction. So what is HER2 positive breast cancer? HER2 positive breast cancer is a breast cancer that tests positive for a protein called human epidermal growth factor receptor 2, or you could call it just abbreviated by HER2, which promotes the growth of cancer cells. In about 20 to 30% of breast cancers, the cancer cells are caused by overexpression of the HER2 gene, which tends to make the breast cancer cells a lot more aggressive and invasive. Okay.
Yeah, if you speak too fast, you feel very annoying. Just didn't want to listen.
Okay. I will slow down a bit. Treatment that targets HER2 are usually very effective nowadays. These treatments include trastuzumab, lapatinib, pertuzumab. They all work on different receptors, which I will explain later, and other specific targeting antibodies. However, about 35% of patients with HER2 positive breast cancers are inherently resistant to trastuzumab, which they will not respond to trastuzumab, this drug, at first. And about 70% of patients will acquire trastuzumab resistance within a year. So what is the HER2, the human epidermal growth factor receptor 2? So the human epidermal growth factor receptor 2, or we could call it HER2, is also called ERBB2 or CD340, where the symbol ERBB is the gene symbol. HER2 belongs to the EGFR ERBB family. This family contains 4 receptor tyrosine kinases, so including ERBB1 to ERBB4. The EGFR ERBB family are essential in mammalian development, and gene disruption models in mice reveal that they are critically involved in the development of multiple organ systems, including the brain, skin, lung, and gastrointestinal tract. For example, ERBB2 and ERBB4 knockout mice, which means they don't have ERBB2 and ERBB4 genes, die at mid-gestation because of deficient cardiac function associated with a lack of myocardial ventricular trabeculation and display abnormal development of the peripheral nervous system. So HER2 is a kind of HER2.
Do I need to give you a pacemaker to slow down? You're too fast.
So, okay, where was I?
You see, even yourself got lost, right?
Okay.
You drive too fast.
Oh yes, so HER2 is a kind of receptor-like tyrosine kinase. The structure consists of an extracellular ligand binding domain, a transmembrane domain, and an intracellular tyrosine kinase domain. They dimerize upon ligand stimulation and transduce their signals by subsequent autophosphorylation catalyzed by receptor cytoplasmic tyrosine kinase activity, which results in an array of downstream signaling pathways. So importantly, HER2 and HER3 singly expressed in cells without other ERBBs cannot be activated by ERBB ligands, even at very high-level ligand concentrations. Subsequently, partner selection appears to be a key signaling activity among HER proteins and their signaling functions, follow a distinct order favoring heterodimers over homodimers, So HER2 has the strongest catalytic kinase activity, and HER2 containing heterodimers have the strongest signaling function. HER2 and HER3 are shown to be obligate partners, which means they will always bind together, but the complex forms the most active signaling heterodimer of the family and is essentially for many biological and developmental processes. With HER2 gene, it is homologous to the new gene in rodents, which was discovered prior to the HER2 gene. So without mutations, the new gene can introduce transformation of cells which are considered the first steps of tumorigenesis. While the homologous gene in rodents, the new gene appears to acquire mutational activation for tumorigenicity. Human HER2 appears to hold tumorigenic potential through overexpression alone, which means that it doesn't need a mutation. HER2 amplified breast cancers have biologic characteristics that distinguish them from other types of breast cancers. These include increased sensitivity to certain cytotoxic chemotherapeutic agents, resistance to certain hormonal agents, and increased propensity to metastasize to the brain. HER2 overexpression and amplification is also seen in subsets of gastric, esophageal, and endometrial cancers.
You have gathered a lot of information, but still stick on one slide, didn't move. It will be good during your presentation. You can put some figure to help you explain the sentence. It'll be better. Otherwise, just keep reading the manuscript. It's not helpful for the audience.
Yes.
Okay?
But I have another.
But in the previous slide, stick on the same slide for quite a long period of time. And you describe a lot of information. Okay? I'm sure everyone got lost. Right? I know you prepare a lot of information, it's good. But you have to make it become readable. Right? Not just only a sentence, maybe just do a small drawing so we can check the drawing, then combine it with your reading. So that will be wonderful for us to understand. Otherwise, I'm sure we will get lost in the first 2 minutes. Okay?
I understand.
Right. And also slow down.
Okay.
Don't be nervous.
Okay. So this picture shows that upon ligand binding to their extracellular domain, HER proteins undergo dimerization. So in this picture, in this case, it's HER2 and HER3 dimers.
You use the cursor to point out which component you want to describe, and the signaling process pathway at which point you want to tell us, so we can follow the figure. Otherwise, just give us a big figure, a lot of content. We don't know which part. Again, we will get lost.
Okay.
Okay?
So in this case, the HER2 and HER3 heterodimers. So upon ligand binding to their extracellular domains, here, HER protein undergo dimerization and phosphorylation of their intracellular domains. These phosphorylated tyrosine residues dock numerous intracellular signaling molecules, leading to activation of a plethora of these downstream second messenger pathways, and the crosstalk to transmembrane signaling pathways, leading to diverse biological effects. So ligand binding induces a conformational change in their extracellular domain. So it does not previously look like this. To the extracellular domain that induces the active conformation and promotes the dimerization and consequent transphosphorylation.
Yeah, the information is good, but remember, main point of this paper is not a mechanism of the HER2 signaling pathway. HER2 is just only the model antigen for its ulcer to do the immunotherapy. Okay? So just briefly go through the pathway. Maybe you don't have to go through the pathway. Just let us know, yeah, HER2 is an important component in this pathway, and it's kind of reset. That's it, that's enough. The point is we want to study using this T cell to target this receptor to kill a specific cancer cell. Okay, that's our main point, right? So we just described a lot of other things. So we create a lot of branch. So we cannot concentrate on the main thing of this paper. Right? So we just got lost.
But these signaling pathways are for later use for the trastuzumab and its internal individual.
Yeah. The trastuzumab is a monoclonal antibody bind to this receptor, right? They
Yes.
will block their signaling pathway. Right?
Yes.
But here they want to combine this antibody. This antibody recognizes HER2, and the tumor cell expresses a lot of HER2, right? But usually the killing is not efficient, right? So now they put extra T cell into the patient or animal to enhance the killing effect of this antibody. So that's our main fault of this paper, right? Can you understand that?
Yes. Second.
So that's not our main point. We don't want to discuss their pathway in this paper. But we know that, yeah, they have this pathway. And now we have a drug, an antibody drug to block this target and cause the death of the cancer. But usually it's not quite efficient. So now they think about an alternative treatment combined with this antibody to enhance the killing of this drug. Okay? That's the important thing.
So do I need to give like more information on like how trastuzumab like blocks the pathways, because it might be useful for later for trastuzumab resistance, and how the gamma delta T cell can like bypass the resistance mechanism.
The title is immunotherapy. The pathway is well known. And the drug, the point is we use this antibody to direct gamma delta T cell to kill this cancer cell. Okay?
Yes.
Still, the signaling pathway is not a main part. So you should not spend so much time on this corner and this corner. You have to spend more time on the other part. Okay?
Yes. Exactly.
Right. So keep going.
Should I like skip over some of the?
Yeah, you skip over this part.
Yes. I might have to skip over a lot of parts.
No, why? You said something, let me check.
So, okay. So, ERBB2, the HER2 overexpression hyperactivates components of the cell cycle machinery, changes which are also linked to resistance against apoptosis, including therapeutic agents. So, overexpression of HER2 in human mammary, human epithelial cells induce proliferative advantages, transformed characteristics, tumorigenic growth, and in 3D models induces proliferative and anti-apoptotic changes that mimic early stages of epithelial cell transformation. However, neither mutations within the transmembrane domain or the extracellular domain of HER2 have ever been reported in naturally occurring human cancers, and despite these experimentally acquired mutations, Human breast cancers appear to be always characterized by overexpressing of wild-type HER2. And more evidence suggests that HER2 amplification is an early event in human breast tumorigenesis. HER2 amplification is seen in nearly half of all in situ ductal carcinomas without any evidence of invasive diseases. Also, HER2 status is maintained during progression to invasive disease, nodal metastasis and distant metastasis, indicating the correlation with cancer metastasis. So, should I skip this?
No, you say something about it in the chat.
Yeah, okay. So overexpression of HER2 can change the composition of her family dimers, significantly increasing HER2 containing heterodimers and HER2 homodimers. So homodimers and heterodimers. These increases can deregulate cell polarity and cell adhesion. Increased HER2 homodimers disrupt cell polarity, which is the hallmark of many epithelial cancers. So increased HER2 and HER3 dimers drive proliferative, survival, invasive, and metabolic functions. It activates P13K/AKT. This pathway, the activated PI3K/AKT pathway also triggers the mTOR, which is not shown in this picture. The mTOR, a master positive regulator of cell metabolism. It also increases signaling of the SRC kinase. Evidence suggests that SRC kinase are important second messengers of HER2. They are potently transforming and tumorigenic when constitutively activated, and their activation is seen in many human tumor types, including breast cancers with or without HER2 overexpression. So increased total expression of HER2 carries with it an increased expression of subsets of HER2, including nuclear HER2 and the rare delta HER2 isoform. And these increase in each of these is functional relevant to cell transformation. In addition, HER2 activation can activate the RAS and the RAF MEK pathway. So this one here, which favors cancer cells growth and migration. So let's talk about the drug used in this research. So the trastuzumab, or sold under the name of Herceptin by the Roche company, is a recombinant humanized monoclonal antibody directed against the extracellular domain 4 of HER2. It was the first Food and Drug Administration approved targeted therapy for breast cancer. Clinical studies have shown that the combination of trastuzumab with standard chemotherapy produces far better response rates than does chemotherapy alone. Thus, the combination that includes trastuzumab have been considered as the standard care of HER2 overexpressing breast cancer patients. However, the in vivo mechanism of trastuzumab are not fully understood, though there are several suggested mechanisms based on experiments. So the first suggested mechanism in the HER2 like trastuzumab is the HER2 degradation. So the trastuzumab have been proposed to trigger HER2 internalization and degradation through promoting the activity of tyrosine kinase, ubiquitin ligase CCBL. It was observed that the binding of trastuzumab to HER2 recruits CCBL to its docking site, where CCBL ubiquitinates HER2 and leads to its degradation. However, how CCBL is induced as trastuzumab binds to HER2 and how CCBL mediates HER2 degradation remains unknown. And the second and most well-known effect of trastuzumab is the inhibition of the MAPK and PI3K AKT pathways, which leads to an increase in cell cycle arrests and the suppression of cell growth and proliferation. It is widely accepted that by interfering with the dimerization of HER2, trastuzumab inhibits HER2 activation and suppresses AKT phosphorylation. It is also shown by experiments that trastuzumab, by binding to HER2, can block tyrosine kinase SRC signaling and thus increasing PTEN level and activity. So the PTEN acts as a tumor suppressor gene through the action of its phosphatase protein product. This phosphatase is involved in the regulation of the cell cycle, preventing cells from growing and dividing too rapidly. This also results in the suppression of PI3K AKT signaling and reduction in cell growth and survival. And the third mechanism, the stimulation of ADCC, the antibody-dependent cellular cytotoxicity responses to drugs. As an antibody, one of the major mechanisms of trastuzumab is to attract immune cells to tumor sites that overexpresses HER2 by a mechanism called antibody-dependent cellular cytotoxicity, the ADCC. It is shown that natural killer cells could target HER2 overexpressing cells coated with trastuzumab via a CD16-mediated ADCC mechanism. So, in brief, NK cells bind trastuzumab to HER2 positive cancer cells through an special Fc receptor. Upon binding, NK cells are able to induce cancer cell death by releasing lytic enzymes. Trastuzumab triggers antibody-dependent cell-mediated cytotoxicity principally by activating Fc gamma receptor on NK cells. In a particular study, researchers showed that Fc gamma R3A158 genotype along in combination with another Fc gamma receptor 2A genotype was significantly associated with better response rates and progression-free survival to trastuzumab compared with other Fc gamma receptor genotypes. This study supports the hypothesis that Fc gamma receptor polymorphisms play a role in trastuzumab-mediated ADCC and predict responses to trastuzumab. So, in this figure, on the right figure, this one, trastuzumab binds to the extracellular domain 4 of HER2. So there are 4 domains in the extracellular space, and trastuzumab binds to the fourth one. And also, the notable one is trastuzumab that binds to the second one. And this binding inhibits all downward cascades of AKT, mTOR, RAS, RAF, and MEK. This also shows that it disrupts SRC signaling, and with specific Fc gamma receptors, NK cells can bind to tumor cells and trigger ADCC. So this graph also shows that besides HER2 inhibitors, there are also other drugs, like the PI3K inhibitors or like other drugs that inhibits this cancer in different ways. However, we will focus on HER2 inhibitors, the monoclonal antibodies in this paper. So even though trastuzumab has significantly improved outcome in breast cancer patients, less than 35% of the patients with HER2 positive breast cancer initially respond to trastuzumab, which means these patients are primarily or inherently resistant to the drug. On the other hand, about 70% of patients who initially responded experienced progression to metastatic disease within a year, suggesting that secondary or acquired resistance to trastuzumab frequently develops. The mechanism of trastuzumab resistance is summarized into several categories. So the first mechanism is the stirring effect. So HER2 can mutate to cut off its extracellular domain through a process called proteolysis, which inhibits the binding of trastuzumab to the receptor. This process generates the truncated P95 HER2 isoform, which with constitutive kinase activity, which promotes the continuous activation of oncogenic signaling and bypasses trastuzumab effects. The second mechanism is the overexpression of other tyrosine kinase receptors. So in the here and here. So since trastuzumab does not prevent HER3 dimerization, the overexpression of HER3 could overcome trastuzumab-mediated inhibition of HER2 signaling. It was previously found that co-expression of HER2 and HER3 is required for the transformation of NIH3T3 cells, and that HER2 and HER3 dimerization results in increased tyrosine phosphorylation of HER3, which subsequently increases PI3K recruitment and downstream activation. These findings imply that cancer cells, by utilizing HER3 signaling, can compensate for the inhibition of HER2 signaling mediated by trastuzumab. Also, on the top left corner, you will see a c-Met is a critical oncogene who is critical in some of the organ regenerations and physically interacts with HER2 and synergizes with HER2 signaling to confer resistance to trastuzumab. This implies that c-Met overexpression is critical for treatment-acquired resistance to trastuzumab. And the third mechanism is summarized as intracellular research showed that patients without PTEN, over here, number 3, without PTEN have overall lower response rate to trastuzumab. Also, patients with PI3KCA mutations have shorter progression-free survival than did those without mutations. Another main reason for less response to trastuzumab is the SRC activation. So recall that the SRC is in-cell signaling that progresses the tumor. So with failed attempts to inhibit SRC signaling, it maintains its tumorigenic effect. However, several studies have demonstrated that trastuzumab resistance isn't caused by decreased ATCC. So none of the above is causing the decrease in ATCC. So we can use the gamma delta T cell that I will introduce later. So the human gamma delta T cells. So gamma delta T cells are important subsets of unconventional T lymphocytes, which they are very scarce in the human body, as they have the ability to recognize a broad range of antigens without the presence of major histocompatibility complexes, the MHC molecules. They can attack target cells directly through their cytotoxic activity or indirectly through the activation of other immune cells. Gamma delta T cells function responses are induced upon the recognition of stress antigens, which promotes cytokine production and regulates pathogen clearance, inflammation, and tissue homeostasis in response to stress. There are two subsets of gamma delta T cells, the V delta 1 and the V delta 2, that always associate to the V gamma 9 chain in adults. The majority of gamma delta T cells do not express CD4 or CD8, but harbor a V gamma 9 V delta 2 TCR specific phosphoantigens. Both human gamma delta T cell subsets exhibit a cytotoxic potential that is induced through the expression of cell surface receptors. The gamma delta TCR is T cell receptor and NKG2D, the natural killer group 2D, and is mediated by the release of soluble mediators such as perforin or granzymes. They can produce granulysin, which is a potent antimicrobial proteinated express ligands such as CD95L and tumor necrosis factor related apoptosis inducing ligands, which engage several death receptors on target cells. In addition, they can kill the targets indirectly through antibody-dependent cellular cytotoxicity, the ADCC mentioned above, and in a CD16-dependent mechanism. So So to elaborate, different receptors, namely the TCR, co-stimulatory molecules in NK receptors play a key role in regulation of gamma delta T cell mediated immune responses. For instance, the activation of blood V gamma 9B delta 2 T cells by anti-NKG2D antibody or its ligand from tumor cells, the MICA-B MHC class 1 chain-related sequence A or ULBPs induce TNF-alpha production and the release of cytolytic granules. Nonetheless, phosphoantigen stimulation of V gamma 9B delta 2 T cells is able to induce TNF-alpha production through the upregulation of CD16 expression. Its role in mediating ADCC was highlighted using therapeutic antibodies such as anti-CD20 rituximab and anti-HER2 antibody trastuzumab. And another important role of the gamma delta T cell is the promotion of a robust NK cell-mediated anti-tumor cytotoxicity. It has also been described through CD137 engagement on blood-activated gamma delta T lymphocytes, which in turn induces the upregulation of NKG2D NK cells, followed by the eradication of tumor cells. So the gamma delta T cell combined immunotherapies. So researchers have previously amplified V gamma 9 delta 2 T cells in vitro from neuroblastoma patients by using a synthetic phosphoantigen, the bromohydrin pyrophosphate. Proliferation was observed after single in vitro stimulation with BRHPP, the bromohydrin pyrophosphate. After 21 days in culture, V gamma 9 delta 2 T cells represented more than 80% of cultured cells, a 50-fold expansion from baseline. Moreover, these V gamma 9 delta 2 T cells were able to lyse allogenic and autologous neuroblasts, which is a gamma delta T cell receptor-dependent cytotoxic activity. In addition, anti-osteolytic drugs such as amino bisphosphate notase activate the same lymphocytes by inducing endogenous PAG, the phosphoantigens, and could thus have related therapeutic applications. Also, there are several studies that prove the increase of lytic activity by gamma delta T cells binding with monoclonal antibodies such as trastuzumab. However, the in vivo outcome of the gamma delta T cell combined with trastuzumab remains untested and will be shown later in this study that it does improve the trastuzumab bioactivity in the tumor xenograft model. So into the results. The experiment is roughly divided into 3 portions. The in vitro testing of the V gamma 9 delta 2 T cell and it's combining with trastuzumab. The second part is the in vivo testing. And the third part is after the in vivo testing, they will take out the tumor and put it in a flow and see if the immune cells are infiltrating the tumor cells, indicating that it is working. So in this experiment, the V gamma 9 V delta 2 T cells are endowed with an NK-like HLA unrestricted cytolytic activity, meaning that they now do not possess the property of a cytotoxic T cells, which was the HLA restricted ones, but rather the property of natural killer NK cells. They are to evaluate the cytotoxicity of V gamma 9B delta 2 T cells with trastuzumab. The HER2 high, meaning that they contain a lot of HER2 proteins and express a lot of HER2 genes. That this cancer cell line, the SKBR3 and the HER2 low, meaning less HER2, the breast cancer cell line of MDA and B231, they were used as allogenic target cells on the left panel of A. We see the cell surface expression by the HER2 marker of the SKBR3, so the left one is SKBR3, the HER2 high cell line. So we see that with the dashed line as isotype control, we are able to confirm that the SKBR3 cell line used indeed contain high amount of HER2 protein, just to make sure. On the right panel, with isotype control, we are able to confirm that the MDA MB231 cell line used indeed contain lower amount of HER2 protein. The SKBR3 cell line was used to represent HER2 positive tumor cells which overexpresses HER2 proteins, HER2 genes, and the MDA MB231 cell line was used to represent the HER2 negative tumor cells. The first two panels are done with the help of antibodies and flow cytometry, which will be discussed in the next slide. So the flow cytometry. So on the right hand side, this picture is like basic information about the flow cytometry fluid system. So the flow cytometry contains three main systems: the fluid system, optics and detection systems, and the signaling and post-processing systems. So here, the fluidic systems consists of a central core through which the sample fluid is injected, enclosed by an outer sheath fluid due to narrowing of the sheath. So the sheath fluid comes from two sides, and thus, using hydrodynamic focusing, they focus the cells into a single file of cells. So like this may be in a cuvette or in a nozzle. So the fluid velocity by the hydrodynamic focusing, the velocity increases. So the sample is introduced into the center and is focused by some other effects. So this allows the creation of a stream of particles that is in a single file, so we won't have overlaps or like doublets that are very hard to detect and differentiate. So the fluorescent stain of a single cell line of cells are then activated by the laser light. In this case, the fluorescent staining used in this research are FITC and Pacific Blue. The fluorescence emitted by the stains will be gathered by photomultiplier tubes after being sorted by specific dichroic mirrors. The flow cytometer is essentially a high-tech fluorescence microscope that can differentiate cells by their fluorescence labeling size and cell structure. So like right over here, we have laser light coming inside and by activating the cell, we produce a forward scatter and a side scatter and some other fluorescence lights. So the high angle scatter, the side scatter will represent cell granularity and cell structure. And the low angle scatter, the forward scatter, will tell us about the cell size. So we can later make a double histogram or a single parameter histogram to like introduce the data. So this is a typical setup of a flow cytometer, which different dichroic mirrors are placed at strategic points that will reflect the lights and thus separating them. The dichroic mirrors will differentiate different nanometers of light, wavelengths of light, and thus separating them. So in this panel, we talk about the confirmed, like with confirmed SKBR3 cell line and MDA MB231 cell line. We now do an in vitro cytotoxicity assay. So on panel B, the two kinds of cells were individually tested by three different combinations of gamma delta T cell and other antibodies, and also different ET ratios. The ET ratios on the x-axis indicates effector to target ratio, with the different gamma delta T cell combinations as effectors and the cells as targets. The y-axis indicates percentage of cells being lysed specifically by the combination of gamma delta T cell and other antibodies. This is measured by the standard 4 hours chromium-51 release assay, which we will discuss later. The combination tested are gamma delta T cell only, and the gamma delta T cell combined with trastuzumab, and the gamma delta T cell combined with human immunoglobulin G, or the IgG. The combination of gamma delta T cell and the HIGG is considered as the control group. So on panel C, the mean of chromium-51 release assay of the two kinds of cells were measured individually in the presence of trastuzumab only. Gamma delta T cells and only, and gamma delta T cells plus human IgG, and gamma delta T cell plus trastuzumab, where human IgG plus gamma delta T cells also serves as control group. So in the C panel, the y-axis here shows the percentage of cells being lysed specifically by the combination of gamma delta T cells and some other antibodies like trastuzumab and human IgG. So the left panels of B and C are conducted with SKBR3 cells, and the right panels of B and C are conducted with MDA and MB231 cells. With left panels of B and C, we conclude that in the presence of gamma delta T cell, the cytotoxicity of trastuzumab against HER2 high SKBR3 cells increases significantly, in contrast to MDA and B231 cells on the right, where in the presence of gamma delta T cells, the cytotoxicity of trastuzumab against HER2 low cells are only increased by a marginal effect, confirming that the cytolytic enhancement of trastuzumab and gamma delta T cells from SKBR3 cells did result from a better HER2 targeting. So this is a generalized protocol for standard 4 hours chromium-51 release assay. So this assay are commonly used for the precise and accurate quantification of cytotoxicity, particularly in the study of tumor cytolysis. This test has the advantage of requiring only very few cells. For example, below is a typical protocol like this. And to do a chromium release assay, you first incubate your target cells with chromium-51. So low target cells with chromium-51, and then you wash the target cells, and then you incubate the target cells with effector cells. So now the target cells will be, so when the when the effector cells kill the target cells will already have chromium-51 in them as a label, as a marker. So when the effector cells kills them, they release the chromium-51. So by the quantity of the chromium-51 label and the marker, we can know that how much cell have died from this effector cell. So the conclusion of the first experiment. So with these observations in mind, we reach the conclusion of the first experiment that trastuzumab combined with gamma delta T cells strongly increases the HER2 targeting and killing of HER2 high SKBR cells. I didn't finish the others. So
Right. You spend too much time on the small thing. You should focus on the main thing that you spend. Right?
yes.
Really create a lot of range. I know every information is important, but depends on the situation. Currently, for this paper, we can focus on antibody interact with the HER receptor and the ADCC mechanism, right?
Yes.
So, for instance, about gamma delta T cell, you don't have to describe too much about NKG2D response, because here they only use the CD16 receptor on the gamma delta T cell to bind to the receptin antibody. Okay? So that's the main function of the gamma delta. I mean, the gamma delta is not to spend too much. A lot of information about gamma delta. Here, we only need a few items. And also the result part, the mechanism of the killing method use the chromium isotope, which presented at the beginning. Then we know roughly how to do the experiment. Then you introduce us to the result. Okay? And also you describe the method, you use the chart and write a lot of writing on the slide, we can understand it. For the chromium assay, you can find a simple drawing that tell us we can easily catch it. Yeah, you show us this, I will not read it. If I am a real audience, I will not read it. Okay, so that doesn't help. Right? Did you get it? Yeah. And also, that's a presentation. So, but it sounds like you are reading a book for us. You just keep reading your manuscript. But we have a slide, and the slide for you to help us to understand the paper. So every time when you use the slide, it looks like you didn't use the slide, just show us the slide, that's it. You have to use your cursor, then point out which point you want to introduce, okay? So then we can follow. Otherwise, just listen to your reading, that's it. And during the reading, there's a lot of important keyword in the sentence. If you are not an expert, you are a general audience, I'm sure you cannot understand it. So in the case, looks like your presentation doesn't help, doesn't transfer information. At the beginning, we expect you read this paper, so you know it. So you need to rearrange the information and concentrate. Okay? Give us a brief presentation. You don't need so much detail. That's too much. We cannot take it. Okay?
But if the detail is like way too less, with way too less detail, will like the audiences not know what I'm doing?
No, you have to decide. Think about it. If you are an audience and you sit here, you only have 40 minutes to present this paper. If you present in this way, I'm sure, give you 1 hour, you still cannot finish. Okay? And you have to remember your audience, they are not experts. And also, they cannot catch so many spatial terms. So we need to use a lot of feedback to help us. Okay? Otherwise, we just got lost in the first 5 minutes. Then you keep reading your book, and that's it. So we didn't learn anything from your presentation. I know you prepare a lot of slides, it's good. And you read a lot of extra citation, that's good. I know you gain a lot of information and knowledge for yourself. So you didn't transfer it to your audience. Okay?
Yes.
So you read a lot, then you need to squeeze this information and somewhat too much detail you remove, then keep some important part, then transfer to us. Because you only have 14 minutes to present. And we want to learn something in these 14 minutes. Okay, and something focused, related to the main part of this paper. We don't want extra information. That would be too much.
Yes.
Okay?
Understand.
Otherwise, you just miss the point. Always depends on the audience and the paper. Okay? If the audience are expert, you can introduce in the other way and use a lot of spatial terms. It's fine, so we can catch it. But remember, your audience, for them, this is a new knowledge. Okay? So you have to think about it. Depends on the audience, right? So now you have to use a kind of easy, brief approach to tell us, oh, we cannot catch it. We just lost.
Okay.
Okay?
Okay.
Right. So any question about,
Yes.
How can the scientists combine the transcriptoma and the T cells?
But it is not elaborated in the paper, and I haven't researched it yet. Yes. Because it's like in the way back of the results. So any other questions?
question. Is it true? So you understand it. Now you ask them question. Because they understand that. So now it's your turn. Ask them question.
But okay.
Or who can tell us to turn on the light? Yeah, it's too dark.